much appreciate your feedback on if and how any of sort of the technology portfolio and questions we're asking could be relevant. I do hope so. So I'm at the Danforth Plant Science Center in St. Louis, Missouri. It's a nonprofit plant science research institute. And my lab is interested primarily on how plants make decisions and interact with the environment. And we focus on root systems, but really it's an integrated view of root and shoot that we're interested in. So here you're looking at a multimodal data set. This is optical imaging, which is a gel-based imaging that allows us to see the root system itself of this maize plant. And in green here, we're using a radioisotope, carbon-11, to watch carbon move in real time from basically the photosynthate move from leaf to roots. And so we're very interested in particular along a genetic axis of how plants make different growth and in particular foraging decisions in the soil. So does a plant needs to decide if it's going to invest carbon in extending a root that already exists or making lateral roots, and of course the vector of that growth itself. And so we've done quite a bit of work, again, genetically trying to understand the differences. Here we're looking at two maize hybrids, two maize inbred lines and a hybrid, and we're able to map in 4D using some computational tools that we built the individual growth of each root in the context of the entire root system. So really understanding how this complexity builds and how genetics drives complexity. And in fact, we were able to show some nice correlation to roots that we dug out of the field via shovelomics with this work. So genetics does have a strong imprint, obviously, on root architecture, and that's something that systems like this are really good for. If you want to know more, you can have a look at that paper. So we do work in the field. The Danforth Center, unfortunately, does not have a field site, so we travel and have a mobile roots trailer, basically. We drive around the Midwest and dig roots. It started with me in 2013 with a pickup truck and a shovel, and we got the hang of it over time, and basically we dig several thousand roots every year with this standard shovelomics method. So not too deep, I don't think I'm passing the threshold here, but again, what this allows us to do is really capture the effects of genetics and genetic variation for root traits at a very large scale. And recently, in collaboration with Andrew Leakey at University of Illinois Urbana-Champaign, we've combined this with very large-scale mini Rhizotron data sets. So this is a drone image and all these white things here. It had been labeled arrows from the gods, which I really like. That makes it sound very dramatic. But there's many thousands of mini Rhizotrons in this tube, and then we dig up the root crowns. So the root crowns give us, obviously, high information content of superficial root systems, and then we get some information at depth with these mini Rhizotrons at scale. And Andrew's been able to analyze these using a machine learning algorithm to segment roots in soil. So the idea here is that we're actually looking for correlations of these different root phenotyping methods and different traits at the genetic level. So this is just an example of a genome-wide association analysis. We're looking over multiple years for various root traits, mini Rhizotrons, root crowns, and even gel imaging, and basically trying to understand are there shared genetics there. That would give us a clue to something that could be impactful. So how do we actually get information from the root crowns themselves? My group's heavily invested in X-ray imaging. As was alluded to before, we have two systems. One is a large-scale industrial system, and one is a microscope that allows us to get cellular and subcellular resolution. And I'll give you an idea of how we're using these two things. Of course, we can use these, and we do, to study roots in pots and various types of soil. This isn't a soil, obviously, but we're getting better and better at this, so field soils are certainly possible, and we can do this 4D analysis now increasingly in realistic substrates. So that's a nice thing, and then of course at the end we get a root model of a living root system, and we can do pretty large things as well. But something else we've been using it for is just to extract more information from what we can get from the field. So obviously if you have these root crowns and you hold it up to a pinboard, you can get some information. If you take a 2D image, which you do a lot of, you can get more. But you lose quite a bit as well. And in about 3 minutes, we can scan and get a three-dimensional reconstruction of the root system. And this gives us a lot of information to work with. So the idea here is we can extract lots and lots of features, This is a study where we correlated it with root pulling force. So you see there's a really nice correlation with some of these 3D root architecture traits with the force it takes to pull the root system out. I mean, that's intuitive, but we're able to quantify it here. Root pulling force obviously being a field-ready technology, let's say. But in the end, all this information allows us to use statistical analyses to really discriminate genotype or genotype by environment interactions. So you can see here our 4 replicates cluster really nicely for these different maize genotypes. So high information content gives us a lot of additional power for discrimination. And that's the key. And then with the X-ray microscope, we're able to do some multi-scale imaging. So here you're looking at individual cells within, this is a sorghum root system scanned by X-ray, but individual cells, these are nuclei here, or the primordia of a nodal root. So we've worked with a lot of genetics that basically are quantitative regulators of nodal root development, and of course very interested in gravitropism and other properties like this, that again, over time do affect the entire root system and certainly rooting depth. And then increasingly we're studying interactions between plant and microbes. So this is a maize root infected with AMF fungi, and I don't know if you can see these bright spots here. These are arbuscules within the cells. We can see the hyphae running between them. It's always difficult to show data like this with the movie, but hopefully you get the idea. If we then segment these structures out, this is very preliminary, but here you're looking at the hyphae colonizing a maize root. And of course, if we can get this image, we can quantify it quite well. Okay, in the final few slides, there's another technology, I guess, that we're developing. I call it fancy boxes. No one else likes that name, but they're not here, so I'm just going to call it that. So this is a novel concept. We're getting some information out that maybe hasn't gotten before. So the idea here is we'd like to capture the entire root architecture, 3D root architecture, and monitor real-time resource fluxes, as well as develop test beds for new sensors. For example, ground-penetrating radar or some electrical resistivity tomography tools that have some potential but need development. So the idea is pretty simple. It's a very large box, and you can build these of any size. But our key innovation is a PVC and fishing line scaffold that goes in the middle of this thing. And I'll show you why that's important in a minute. And anyways, we can collect runoff and basically drainage here. And then we fill this up. This isn't soil, of course. We will eventually move to soil, but for development purposes, we've used turfus and calcinated clay. It works quite well. We fill this up, embed it with a number of sensors. These are water potential sensors. They also measure temperature. And we've also used the Caro gas analyzer to measure CO2. And basically, this allows us to model resource flux over time. There's another reality where these movies might be moving, but you can see we're basically at hourly data. Here you're probably looking at daily data of water and temperature. But at the end, we carefully harvest these things with a shop vac. So that's the key innovation here. We can slowly capture the entire root system more or less as it actually grew. And so here we're looking at 2 different genotypes of switchgrass. So we can clearly see genetic differences. And it's literally the entire root system, essentially freely grown. I mean, a few of these roots have hit the sides or hit the bottom, but it's essentially an entire 3D root system. We can see very clearly the effects of the water stresses. This is grown at -1.5 megapascals, which is a fairly strong stress. Switchgrass does well as a hardy perennial. And then to get the actual 3D models themselves, we use a method called photogrammetry, which is just taking thousands of photographs in a circle with a handheld digital camera. And basically, we get the geometry of each of these, we can align them, and we can generate a 3D model from that. So this is basically the last bit that I'll show you, but this is essentially, we've developed basically a studio for this that allows us, we had to use, it looks a little psychedelic, but we had to use a lot of random patterns and things to help get the model. This is what our studio looks like. This is what a post-process root system would look like once we remove the room, the scaffold, the fishing line, etc. And we're continually sort of building on this. So that's the last bit I'd like to show. show you. So, you know, essentially, I think we're all quite aware of all of these points, but integrative approaches are needed. There's hard trade-offs with virtually every root phenotyping method. Our particular area is focusing on high-content phenotyping, so really trying to maximize the amount of information we get out of a particular sample, and we like to focus on dynamics, and as has been mentioned several times, functional outcomes. So I think there are some real opportunities here for developing sensors, developing infrastructures with sensors and real-time data, and to some extent using some artificial intelligence and machine learning methods to really turn these real-time data streams into actionable information or information about biology. So I think I'll stop there, and thanks for your attention.